Hello, and welcome to the next of our matric revision lessons for life science. This time we are going to be looking at chromosomes and cell division, more specifically meiosis. Now, this section links what you have done this year in looking at the structure of DNA and how it works, and also the human reproductive system. It's kind of a link between the two of those systems. So let's have a look at how they form a link. I'm sure you've seen this before. This is a human life cycle, and I'm sure you've seen that term before as well, because all cells in our body, normal cells, not reproductive cells, all are 2N, and that is called diploid, meaning they have two sets of chromosomes. You get those sets of chromosomes from each of your parents. The one you get from your mother is called the maternal set of chromosomes, and the ones you get from your father are the paternal set of chromosomes. So, in every single cell in your body, you have got two sets of instructions for each characteristic. For example, you inherit a gene for eye color from your father and a gene for eye color from your mother. And in the section on genetics, you probably had a look at how those two genes interact with each other to give you your particular eye color that you have. Now, looking at this, we have the male and female producing sex cells or gametes. And those gametes fuse in the process of fertilization. So, each of these gametes must be haploid. In other words, they must only have one set of chromosomes. Now, you inherit, or you have in your cells, two sets of chromosomes, one from your father, one from your mother, but you can only pass one of those on to your child. So your child will also have two sets of chromosomes governing each characteristic, one from you and one from your partner. And to make sure that this can happen, there is a special type of cell division called meiosis. And that occurs during the production of gametes, which is called gametogenesis. And as we talked about in the section on reproduction, that would occur in the ovaries and the testes. So, meiosis halves the number of chromosomes in your body. Now, let's have a closer look at chromosomes. In grade 10, you looked at the cell and you discovered that in the nucleus of the cell are long, thin threads of chromatin, which is made of DNA. Now, in each one of your cells, your tiny, tiny little cells, there's nearly 2.5 meters of DNA in a tiny little nucleus. So that's quite a lot of very, very thin threads, which is fine when your cell is functioning normally because it's got to read the instructions on those threads. But when your cell divides, things can get a little bit tangled. And that's when chromosomes form. And chromosomes are formed from the long threads of DNA which get coiled up. And you should also know that in humans, we have 46 of these chromosomes. In other words, we have 46 of these pieces of DNA. Now, if we look at this picture, here is the nucleus. And you can see it's chock-a-block full of these chromatin threads. When the cell wants to divide, you can just imagine taking half of what is in here and trying to sort it out. Could lead to lots of tangles. So what happens to prevent the tangles? The long chromatin threads get coiled up. They get coiled around proteins called histones, a bit like if you take string and you wind it around a piece of wood or something like that in order to keep the string nice and neat. And those then get all coiled up to form a chromosome. So a chromosome and chromatin network are basically made of DNA. They're just in a different form for a different reason. Now, you should know that each chromosome consists of 2 chromatids joined by a centromere. Okay, so let's have another look at how this happens. We go from the very thin double helix, wrapped around proteins, that gets wrapped a bit more, that gets wrapped a bit more, and it's basically like taking wool or string and rolling it up into a ball, so it's less likely to get tangled. And that ball of DNA is your chromosome. And the chromosome consists of 2 halves called a chromatid, and the 2 chromatids are joined by a centromere. Now, why sometimes do chromosomes appear single and sometimes they appear double? What's the difference between them? In your cells, you have got chromosomes that are single. They consist of 1 copy of a piece of DNA all coiled up. Those 2 sets of chromosomes, let's say for example there's eye color at that particular point. You got one of these from your mother, let's say the green one came from Ma and the purple one came from Pa, but they would both have instructions for making eye color. Will they necessarily be the same instructions? No, but there will be instructions for eye color. Then what happens? Just before the cell starts to divide, the DNA replicates. And what you see here are the 2 replicated homologous chromosomes. And when a chromosome replicates, it's composed of 2 copies of the DNA, and each copy of the DNA is called a chromatid, and they are joined together by a centromere. Now, let's have a closer look at these homologous chromosomes. Remember, homo means the same. So these are chromosomes that contain instructions for the same characteristics. So you will have 1 chromosome which comes from your mother and 1 which comes from your father. Now, these colors on the chromosomes here are not just to make the chromosomes look pretty. They are saying in that particular place is a particular gene, which is a piece of DNA coding for a particular characteristic. So each chromosome contains many genes. Some genes have a bigger bit of DNA than other genes. Some are fairly short. Now, where a gene is found on the chromosome is called the locus or location of the gene. The other term you need to know for this is the genes that govern the same characteristic found in the same position on the chromosome are called alleles. They are genes controlling the same characteristic. Now, you probably also at class looked at something called a karyotype. And what a karyotype is, it's a photograph taken of all the pieces of DNA, all the chromosomes, as the cell is about to undergo cell division, and then those chromosomes are paired up in their homologous pairs. And as you can see, there are 23 pairs of chromosomes because we've got 46 chromosomes altogether divided into 23 pairs. Now, in 22 of the pairs, the chromosomes are the same. They're the same length, they look the same, and they contain alleles for the same characteristics. Only difference is the 23rd pair. Might not be, the 22 pairs that are exactly the same are called autosomes, and they control everything about your body. For example, what color your hair is going to be, how tall you're going to be, are you going to be good at maths, things like that. The other cells here are the sex chromosomes, and they can be the same or they can be different. Okay, and these are sex chromosomes or the gonosomes, and consists of a large chromosome. Oh, by the way, they're not really pink and blue. They've just been colored that way to help you distinguish the difference between the two. So females have got 2 X chromosomes, whereas males would have 1 X and a smaller Y chromosome. I'm sure you've seen a picture like this before, and that is the karyotype of a human. And if you look at it, you can tell, aha, this is a male. How do we know? If you look at the chromosomes, there's a small and a large chromosome. This is a female because the pair of chromosomes are absolutely identical, so it contains 2 X's. Whereas in a male, the male would contain the large X and a smaller Y chromosome. Sure, I think by now you need a break. So let's take a break, get some fresh air to get your minds working. See you just now. Welcome back. I hope the break refreshed your brain and you're ready for the next section. What sometimes happens is that people don't have the right number of chromosomes. So sometimes there are abnormalities in the chromosome. So let's have a look at one very, very common example and what can go wrong. Why does this happen? Basically, it is if meiosis does not occur properly. And when we look at the detail of meiosis, we will look at exactly why. And this leads to cells having extra or missing chromosomes. Now, most of the time, if a chromosome is missing, the child never gets born because there is a set of instructions missing and the embryo can't develop properly. If there is an extra chromosome, especially if it's one of these small chromosomes, the embryo will survive, but they won't be perfect. There will be something causing a problem because they've now got extra instructions on the DNA, which causes confusion in the cell. Now, a very common example, which you've probably heard of, is trisomy, which means there's 1, 2, 3 chromosomes in what should be pair 21. And when that happens, we say the child has Down's syndrome, which is one of the most common chromosomal problems that there are. Down syndrome children, why do they end up like that? Okay, and that is because when the chromosomes or the homologous pairs should separate during meiosis, they don't. And that is called non-disjunction. The homologous chromosomes do not separate. And that can happen either during the first division of meiosis. Here you can see what's happening here. There should be one of those chromosomes going into that cell. There isn't. Now, we've only drawn one chromosome here, but in reality, if this is human, that means there would be 46 other chromosomes. There would be 46 chromosomes in the cell, so this would have 22 chromosomes, whereas this one would have, 24 chromosomes. That leads to, when the eggs are produced, some eggs do not have a chromosome 21. Other eggs have an extra chromosome 21. Or this can also happen in the second part of meiosis, but again, it's when something goes wrong with meiosis and the chromosomes do not separate. Questions are quite often asked on karyotypes and what can go wrong. So let's have a look at this practice question. This shows chromosomes of a person with Down syndrome, and if we have a look there, we can see the 3 chromosomes. All of the other chromosomes are beautifully in their pairs, except for chromosome 23. Okay, the questions usually start off with fairly straightforward questions to check, do you know your terminology? For example, what is the label for A, which would be homologous chromosomes? Then, how many autosomes? And this checks, do you know what autosomes means? And autosomes are the 22 pairs of ordinary body characteristics, or the chromosomes that control the ordinary body characteristics. Then the type of chromosomes found at position 23, those would be the sex chromosomes or the gonosomes. Then, is this a male or a female? Easy peasy, you have a look there and you can see 2 different chromosomes, so it's definitely a male. Then it says name the type of mutation. Okay, in other words, what's gone wrong here, and this is a chromosomal mutation when there's something wrong with the chromosome. And then you would have to, for 6 marks, describe exactly what goes wrong in meiosis to actually cause a child to be conceived who has Down syndrome. What we're going to look at next is we're going to have a look at cell division. Now, you did the type of cell division called mitosis in previous years, and then this year you have done meiosis. But I'm going to put the 2 of them together so you can see the similarities and the differences between them. So, let's have a look. There are 2 types of cell division, and they occur in different cells at different times and for different reasons. And this is quite important to understanding the 2 different types. Okay, first of all, the one you're familiar with, mitosis, which is something that occurs in all parts of your body to produce new cells for growth, and it occurs in all parts of your body as you grow. And when a cell divides by mitosis, it produces 2 identical cells. And these cells are identical to the parent cell, which, like all the other cells in your body, is diploid. So in mitosis, we are simply duplicating the instructions on the DNA. Meiosis, on the other hand, is very different. This occurs only in the reproductive organs, only for gamete production. And quite importantly, the end result is 4 cells which are haploid, but important is they are different from each other. So meiosis is what causes variation in families. Let's revise mitosis. Not because you have to learn it for this section, but because you need to understand it in order to be able to understand what's happening in meiosis. So, first of all, mitosis, producing 2 identical cells, and it passes through different phases. The cell, before it starts dividing, we say is in interphase. Then it starts dividing, and the preparation for division is called prophase, and that is when the chromosomes replicate. Okay, the chromosomes have replicated and they are now actually forming. The nuclear membrane disappears, and these things here, which are the centrioles or centrosomes, are moving apart, and they are forming what we call the spindle, which is what helps separate the chromosomes during the division. Second stage is metaphase, and in metaphase the chromosomes arrange themselves along the middle of the cell. In anaphase, the chromosomes split, and a half a chromosome or a chromatid moves to each pole. The last stage of cell division is telophase, and that is when the chromosomes arrive at the poles and the nucleus starts reforming. After the nucleus has divided, the cytoplasm divides, so that is cytokinesis, which occurs only after the nucleus has divided properly. Right, let's have a look at these stages, because meiosis has the same stages with the same basic things that is happening, except meiosis, it gets a little bit complicated. So let's make sure we understand the simple mitosis first. Okay, so we start off with interphase, which is the resting phase. In some literature, they will say interphase is part of mitosis. Others say interphase is between mitotic divisions. Then we have the 4 stages of mitosis itself, which is the division of the nucleus. Then it's followed by the division of the cytoplasm or cytokinesis. And the way you can remember them is by IPMATCH. The I stands for interphase, and what happens during interphase is the DNA replicates. So the I can be for in between. Then the P for prophase. You can think of it, that's the preparation phase. And what is it preparing? It's preparing the chromosomes. So it's winding up those long, thin threads of DNA into these chromosomes so that they can easily be sorted out. The next phase is M, and you can always remember M stands for middle, because in metaphase the chromosomes arrange themselves along the equator, which is the middle of the cell. At the end of metaphase, the centromeres divide, and the next phase is anaphase, and that is when the chromosomes split and the chromatids move to the poles. And you can remember this because A stands for away, and the chromatids are moving away from each other. Last phase is telophase, and that is the end one where the nuclei reform and you get back to a normal nucleus, and that's followed by cytokinesis, which is the division of the cytoplasm. Now, you need to understand how these stages work, in which order they go, and basically what is happening in each of these stages, because when we look at meiosis, we're going to build on what we've done on mitosis. See you next time.